Oh, you have to.
You said
Oh, wait, you can do it. Okay, just press got it then.
all right, I'm gonna let everyone in.
Okay, sounds good.
Hi everyone. If you could just mute when you guys come in. Okay, so today I'm going to be doing a lecture on DNA, RNA, and proteins. My name is David, if you haven't attended one of my lectures before. So I just wanted everyone to know that we are recording these lectures, so if you don't want your name or your picture in the recordings, if they happen to be in there, you can change your name or your picture so it's not included. We are recording the lectures and then posting them on YouTube. And this is just some information for you about the audio and video recording of the lectures. So just a brief description on the course. We are doing a 15-week MCAT program. We have lectures on Thursday, Friday, and Saturday at 8:30 p.m. EST, and then every Sunday at 8:30 also. We do office hours if you want to ask us any questions or go over any problems you might have. And then here's just some links. If you haven't joined the GroupMe chat, you can join and post any questions or problems you might have on there. And then I also provided the YouTube link if you want to watch the live recordings, or we also edit the recordings into smaller videos. If you want to just go over a specific topic, you can watch that video. And then we also have our website, socraticmed.net. If you want to go on there, we'll have videos on there also, and all the information about the programs on there. And then we also have an Instagram at socraticmed _ if you want to follow that, and they'll post any events or MCAT questions on there also if you want to follow them. So the learning objectives for today, I'm going to go over the central dogma of biology, and then DNA, the functions of DNA, the structure, and how we replicate DNA. And also RNA transcription and translation, and then finally protein function and structure also. So how does DNA, RNA, and protein relate to each other? So there's this theory called the central dogma of biology. It describes how the flow of genetic information moves from DNA to RNA and then finally proteins. So at first, scientists didn't know which molecule was responsible for our genetic information. They knew it was either DNA, RNA, or protein. So they conducted experiments and they discovered how all three of these molecules were related. So they found out that DNA has a unit called a gene, which encodes for specific RNA, and then that RNA will translate into a protein. So DNA is our genetic information, and then through transcription into RNA, we are able to make proteins after translation. So that's just how DNA, RNA, and protein are related to each other. So the first molecule we're going to be going over is DNA, otherwise known as deoxyribonucleic acid. So like I said before, DNA is the genetic information in all organisms except for viruses that sometimes use RNA. So most organisms use DNA to store their genetic information, and that genetic code is used to make proteins that all our cells in our body use to function. So DNA is replicated and then also inherited from one generation of cells to another. Our DNA specifically is distributed among 46 chromosomes into 23 pairs. Other organisms will have a different number of chromosomes and different amount of DNA. But for humans, it's 46 chromosomes and 23 pairs. So DNA on each chromosome is made up of separate genes that transcribe into RNA and then finally translation into proteins. And DNA is double-stranded, unlike RNA, which we'll go over in the future. RNA can be either single-stranded or double-stranded. So the structure of DNA. So DNA is a polydeoxyribonucleotide. So that is made up of single monodeoxyribonucleotides. So on the right is the structure of a monodeoxyribonucleotide. So DNA uses deoxyribose as the 5-carbon pentose in the structure. So it's a ribose that instead of a 2' OH, it's replaced by a hydrogen instead. So this right here was, instead of an OH, it was replaced by a hydrogen. So that's why it's called deoxyribose, while RNA is called ribonucleic acid because the OH is still present at that 2' position instead of being a hydrogen. And so the monodeoxyribonucleotide is made up of a nitrogenous base. There are 2 different kinds of nitrogenous bases. So the first is purines. Purines have 2 rings, and the 2 purines in DNA are adenine, the symbol for that is A, and guanine, which is G. So those are the 2 purines. They have 2 rings in their structure. And a mnemonic to remember that is pure as gold. So purines, pure, and then as gold. So the symbol for gold is Ag. So you can remember adenine and guanine. Ag is a purine because purine is pure as gold. So those are the purines. And then pyrimidines, those only have 1 ring, and it's cytosine, which is C, and then thymine, which is T in DNA. And then RNA, instead of thymine, they'll use uracil. So cytosine, thymine, and uracil are the pyrimidines. And then those only have 1 ring. So those are the 2 different kinds of nitrogenous bases. So what happens when you bind the pentose to the nitrogenous base? You get a nucleoside. So when you bind a 5-carbon pentose to the nitrogenous base, so the nitrogenous base is covalently linked to the 5-carbon pentose at the C1 position. And then on the right, I also have the structure of the 4 deoxyribonucleics for DNA. So, and then finally, it forms a nucleotide when you join a nucleoside with either 1 or more phosphate groups attached at the C5 position of the carbon. So when you attach a phosphate group to a nucleoside, you get a nucleotide. And nucleotides have a high energy because of the negative charge repulsions of the phosphate groups. So phosphate groups are negative, and when you put two negative groups close to each other, they repel each other, and the negative charge repulsion is the reason that nucleotides have high energy. So molecules like ATP that you know, those are a nucleotide because so ATP, they have high energy because of the phosphate groups in the structure, and the negative charge repulsion generates that high energy. So there's the Watson and Crick model of DNA. So their experiments, they found that DNA is a double helix structure. So they have two linear chains wound in spiral. So like I said, DNA is double-stranded. So those two linear that double chain strand of two linear chain, the double chain strand of two linear chains are wound in spiral, and it forms a helix-looking structure. And the strands of DNA are anti-parallel. So one goes in the 5' to 3' position, and then the other strand goes in the 3' to 5' position. And so in the model, they found that the sugar and phosphate are on the outside of the helix, while the nitrogenous base points on the inside of the helix. And the model also shows that there's complementary base pairing. So the adenine binds to a thymine via 2 hydrogen bonds, while the guanine binds to a cytosine via 3 hydrogen bonds. So since the guanine and cytosine has 3 hydrogen bonds, that base pair is stronger than the adenine and thymine bond. And then the hydrogen bonds and hydrophobic interactions of DNA provides stability to the structure. Any questions on the material I just went over before we dive into a question? Okay. All right. So the first question, which base pair requires the most amount of energy to break? Let me start a poll. Okay, I'll give you guys a few minutes for this. Yeah, so all answers are anonymous, so just give it your best guess. Okay, I'll give everyone about 15 more seconds to do the question. Just put in your best guess if you don't know, or click the last choice. And sorry, yeah, Janelle's also here today with us. If you have any questions, you can put it in the chat and she'll try to help you out with it. Okay, so I'm gonna stop the poll. So most people put D and some people put A. So D is the correct answer. So it's D because guanine and cytosine, they have 3 hydrogen bonds between them, and that will take more energy to break than choice A, which only has 2 hydrogen bonds between them. And then choice B and C, those are improper base pairing, so they would not be one of the choices for breaking it. They would be improper base pair that would be corrected by DNA repair. Any questions on this problem? Sorry, let me get rid of this mark that I made. Okay. All right. If no one has any questions, I'll continue. So there's a rule called Chargaff's rule. So that's the rule that says that the total amount of purines has to be equal to the total amount of pyrimidines, which makes sense since a purine all binds with a pyrimidine. So adenine, which is a purine, binds with a thymine, which is a pyrimidine, and then guanine, which is also a purine, binds with cytosine, which is a pyrimidine. So since each base pair has 1 purine and 1 pyrimidine, the total amount of pyrimidines equals the amount of purines. So if you know the amount of purines or the amount of pyrimidines, you'll know the amount of the other. So, and then also all the total amount of adenines has to equal the total amount of thymines, while the total amount of guanine has to equal the total amount of cytosine, since they base pair with each other. So this is just a mini table that shows you how the total amount of purines and pyrimidines has to equal each other. So more into the structure of DNA. So DNA has a sugar phosphate backbone. So the sugar is made up of alternating the sugar phosphate backbone is made up of alternating sugars and phosphates. That's because the base pairs, the nucleotides are joined by phosphodiester bonds. So the 5' end of one molecule of nucleotide has the OH or phosphate bonded to C5. Sorry, this is wrong. It's supposed to be C3 of the sugar. Nope, sorry. Yeah, so the first nucleotide will have a 5' end, and it will have a oxygen or phosphate bonded to that carbon. The carbon of the sugar. While the next nucleotide on the 3' end, it'll have a free OH on carbon number 3 of that sugar. And then these are the two parts that bond to each other through the phosphodiester bond. So it's a 5' to 3' phosphodiester bond. So the 5' phosphate of one sugar binds to the 3' oxygen OH of the next. So nucleotides are joined by 3' and 5' phosphodiester bonds, and like I said, the 3' carbon of one sugar bonds to the 5' phosphate of the next sugar. And then, so eukaryotic chromosomes, they organize the DNA into either heterochromatin or euchromatin. So heterochromatin, it's compact and transcriptionally silent. So that's DNA that isn't transcribed or replicated a lot. So when in interphase, you only have a small amount of it, because interphase, usually that's when the DNA is being used. So it'll be most likely euchromatin, which is dispersed chromatin. So that's genetically active chromatin. So this DNA, it's being transcribed a lot. They're making proteins from it. So this, as you can see in the diagram, euchromatin, it's less tightly bonded together. There's more space for molecules to interact with the DNA. While heterochromatin, they're compact, they're all bunched up together, so it's hard for them to be using the DNA, so that's why it's transcriptionally silent. Any questions on this before we move on to another question? Okay, all right, so this question, which of the following choices represents the structure of DNA from least organized to most organized? All right, I'll give everyone a few minutes for this question. Let's start a poll. Yeah, sorry for anyone that's confused by the question. Yeah, technically, you can answer it whichever from smallest to largest. Give everyone a little bit more time. About 30 more seconds. All right, I'm gonna end the poll in a little bit. Okay, so it looks like most of you guys got it right. It was choice C. So deoxyribose is the smallest. It's not. It's deoxyribose. It's just the sugar, the pentose. while nucleoside, that's the sugar bounded to the nitrogenous base. And then nucleotide is a nucleoside bonded to one or more phosphate groups. And then the DNA double helix, it's multiple nucleotides bounded together into a double helix formation. And then a chromatin is the whole DNA double helix organized with other molecules like histones. So choice C is the correct answer. Any questions or confusion about this question? Put it in the chat or just unmute. Okay, all right, I'll move on. So next thing I'm gonna go over is DNA replication. So how do we form new DNA? So for us, DNA is semi-conservative. So that means one of the parental strands is used as a template for the daughter strand. So each new double strand of DNA that we synthesize, one of the strands was from the parent cell. So there is one parent cell in each of the two resulting double-stranded DNA, while the other strand is newly synthesized DNA. So that's why it's called semi-conservative. So it's not all parent DNA and it's not all brand new DNA. It's half parent DNA and then half newly synthesized DNA. And then, so how does DNA replication begin? So the first thing that happens is helicase, the enzyme unwinds the DNA to make two single-stranded DNA strands, and they started at origins of replication. So DNA is double-stranded, and helicase works to unwind the DNA to create two single strands, so the other proteins and enzymes can get to the DNA to replicate it. And then, so the next thing that happens is single-stranded DNA binding proteins. They bind to the unraveled strand after the DNA helicase unwinds them, and this is to prevent reassociation of DNA and degradation by nuclease. So these single-stranded DNA binding proteins, they bind to the two single strands in order to stop them from associating back. So they don't want it to reform a double-stranded DNA. They want to keep it at two single strands. And then the next thing is DNA topoisomerase. So they work to create negative supercoils to decrease stress from supercoiling. So when the helicase unwinds the DNA, it strains the DNA when it unwinds it because it creates stress at the far ends of the DNA. So that's called supercoiling, and DNA topoisomerase works to relieve that stress by nicking either one or two strands at specific spots on the DNA, and this works to prevent supercoiling. It relieves the stress. So instead of one really long strand that's being pushed to the edges when it unravels, it has little nicks in it that are then resealed to prevent supercoiling. And so when DNA helicase unwinds the DNA, it does it at origins of replication, and then this creates replication forks. So you can see on the picture on the right, the fork works in one direction, and that is the direction of the leading strand. So in eukaryotic chromosomes, there are multiple origins of of replication forks because our DNA, eukaryotic DNA, is a lot larger than bacterial or prokaryotic chromosomes. So multiple origins creates multiple leading strands and Okazaki fragments. This is to speed up the process of DNA replication. While bacterial DNA, it's just a single loop of DNA, and there's not much DNA in it, so they only need one single origin. While eukaryotic DNA, which is a lot larger, they require multiple origins and multiple replication forks in order to speed up the process. And like I said before, whenever there's a replication fork, there's a leading strand where the DNA is replicated in the direction of the replication fork. So it's just a one continuous strand of replication, while the Okazaki fragments are created in the lagging strand. So since the lagging strand is facing away from the origin of replication, each time the replication fork moves in the 3' to the 5' direction, or there's multiple Okazaki fragments needed because of the replication fork keeps moving in one direction. So the Okazaki fragments are created each time the DNA is made. So DNA polymerase is the one responsible for creating new DNA. So it reads the parental strand from the 3' to 5' position, while it makes the new strand in the 5' to 3' position. So since the leading strand is always being read in the 3' to 5' position, it just keeps making one long continuous strand. But the lagging strand, since it's anti-parallel, it has to be read multiple times by DNA polymerase. And that's why there's all these mini Okazaki fragments. Okay. But each Okazaki fragment needs a primase to make an RNA primer. So while the leading strand only needs one RNA primer, since it's one long continuous strand, the Okazaki fragments need multiple RNA primers because of how it's made up of many small strands of DNA. So each time a DNA is added, DNA polymerase adds a 5' deoxyribonucleotide triphosphate. So each new nucleotide added is either a DATP, DCTP, DGTP, or DTTP, which represents the 4 nitrogenous bases. So there's 4 different nucleotides added. And each time the nucleotide is added, a new phosphodiester bond is made between the nucleotide that was already added and the new nucleotide that's being added, and a pyrophosphate is released. The pyrophosphate is released because DNA replication is it requires energy, and like I said before, the negative repulsion of the phosphates on the nucleotides, that's the energy needed to form the DNA. So the pyrophosphate is released in order to generate the energy needed to form that phosphodiester bond. And another function of DNA polymerase is it removes the RNA primer that was added by primase. So once it finishes replicating the DNA, it'll remove the RNA primer and replace it with DNA nucleotides instead. So for the lagging strand, it only does this one. For the leading strand, it only does this once, while the lagging strand, it has to do it multiple times. And then on the right is just I like this picture of showing all the things that's happening during DNA replication. Then finally, once DNA polymerase is finished replicating all the DNA ligase seals the ends of the DNA together so it doesn't unravel. So it keeps the double-strand DNA together. But like I said before, DNA polymerase, it can't just add on to, it can't just start replicating DNA. It needs a primer. And since at the end, you can't, at the ends of the chromosomes, the strands of DNA, you can't add a primer to the very ends because there's not enough space for there to be a primer and then to add DNA. So at the ends of the chromosomes, there are things called telomeres. So these are still DNA. They're repeating DNA sequences at the end that don't really code for any genes. They're just there to protect the chromosomes from multiple replications. So they prevent the chromosomes from losing base pairs that actually code for RNA and proteins. So telomeres protect the chromosomes from losing essential genes after each replication, because each replication, the DNA gets shorter since DNA polymerase can't finish the DNA replication at the 5' end. Okay. Any questions on DNA replication? Sorry, I know that was a lot.
Hi, can you hear me?
Yes, hello.
Hi, do we need to know about alternate forms of DNA for the exam, like Z or B DNA?
Sorry, what was that?
Do we need to learn about alternate forms of DNA for the exam, like Z or B DNA?
I haven't really seen them on the MCAT or any questions on it. I know you can, it's like really, there's not much on it. It's not very high yield, but so I know like most DNA is like right-handed helixes, while I think Z DNA, it uses a left-hand helix. So there's not, it's like a left-hand helix, and the major and minor grooves, the distance between them is a little bit different. But I wouldn't say it's like a major yield. It might be, but I haven't seen it yet. I don't know if that answers your question.
Hey, Chris, I have a question. How does the DNA know where to?
Sorry, Opal, you're echoing a little bit.
Sorry. Yeah. How does the DNA know where to begin? You said in the bacteria it's only once.
Sorry, Opal, you're still echoing a little bit.
Go back to that. How would it know where to begin if there's multiple start sites? You said in bacteria there's only one, right? So in eukaryote, how would they know? How does the helicase, whatever, which enzyme? Where would they start since there's multiple origins?
Where would the DNA polymerase know to bind and to replicate the DNA?
All right, which one is binding helicase? Which one binds first, polymerase or helicase?
So helicase, it unwinds the DNA first. Yeah.
So who's binding?
Sorry.
Okay, whatever. I'm not sure if it's an enzyme. Whoever is multiplying this DNA, where does it know where to start? And who is it that's starting? An enzyme? Who is starting this replication?
So helicase starts by unwinding the DNA, but DNA polymerase is the one that's replicating the DNA.
Okay, so where does it know where to start since there's multiple origins?
So yeah, So DNA polymerase, there's it's made up of like a bunch of different molecules.
Okay, I get it. Polymerase 1, 2, and 3, and they have different roles. Okay. I think I know what you're
so in eukaryotic DNA, it's called like alpha, beta, gamma, delta. So those are different kinds, and they do so they bind to the DNA through promoter regions that tell the DNA polymerase to bind there and start replication.
Okay, that makes sense. I was just curious. Yes, I've heard of promoter regions. Okay, thank you. And is that like a special sequence of DNA that the polymerase knows this is where I need to bind? The promoter region? Okay.
Yeah. So those are the regions that don't really code for any like genes, but they just they bind to like transcription factors and the DNA polymerase. Sorry, Opal, did you?
Yes, you got it. Yeah.
Did you need a screenshot of a slide? I think I just read.
The semi-conservative slide, I was going to ask that. You had, yeah, let me take a picture of that one. I often had semi-conservative. Okay, I got it now. Thank you.
Okay, no problem. So does anyone else have any questions before I go into RNA polymerase? Not RNA polymerase, RNA. All right, so I'm gonna go over RNA or ribonucleic acid. So there are different kinds of RNA. So the first one I'm gonna go over is messenger RNA or mRNA. So mRNA, it has the information to make the amino acid, to make the protein. So the mRNA codes for the amino acid sequence that the ribosomes need to create that protein. So it's made like DNA replication. It's made 5' to 3' from the DNA template, and it's done by RNA polymerase in the nucleus. And like I said before, instead of thymine that's used in DNA, mRNA uses uracil instead, so U instead of T. And just like DNA replication, the new strand made is complementary and anti-parallel to the template. But before mRNA can code for the protein synthesis, it needs to undergo post-translational modification before being released to the nucleus and then being used by the ribosome in the cytoplasm for protein synthesis. So the messenger RNA, it has 64 codons. It's a universal and ambiguous code. So eukaryotes and prokaryotes, they both use this mRNA codon. But each codon, even though there's 64, it only codes for one specific protein. Not protein, sorry, amino acid. So each of the 64 codons, they code for one and only one amino acid. So mRNA is monocistronic in eukaryotes. So that means each mRNA molecule so each mRNA molecule translates into one protein. Sorry, that's so each mRNA transcribed is only translated into one protein. But it's polycistronic in prokaryotes. So that means depending on where you start the RNA transcription, it results in a different protein. So since prokaryotes, they usually have a lot less DNA, only one ring of DNA. They only have a small amount of DNA, so they've evolved to be polycistronic. So depending on where you start transcribing RNA from the DNA, it results in a different protein. So like you see in this picture on the bottom of the prokaryotic DNA, when you, prokaryotic mRNA, when you transcribe from one position compared to a different position, these two positions, they create two different proteins, while eukaryotes, each mRNA sequence, it will only create one specific protein. And then the next kind of mRNA, next kind of RNA is transfer RNA, otherwise known as tRNA. So this converts nucleic acid language into what I call amino acid language. So this is the part where you're actually making the protein. So you're using the information from the DNA and RNA and converting it into amino acids. So the structure of tRNA, each tRNA has a folded RNA strand with a 3-nucleotide anticodon. So this means that this anticodon, it's opposite to the mRNA. So the anticodon pairs with the codon on the mRNA. And that's how the ribosome uses tRNA to create the protein, by converting the codons on the mRNA into amino acids that are bonded to the tRNA, and it recognizes which amino acid it needs to use because the tRNA has the anticodon bounded to it. So you can say that the tRNA is charged or activated when it's connected to an amino acid. So sometimes in the cytoplasm, there will be unactivated or uncharged tRNA, so it doesn't have an amino acid bounded to it. But once an amino acid that correlates to the anticodon is bounded to it, it's called charged or activated, and it can be used in protein synthesis. Sorry. Yeah, and like I said before, transfer RNA is in the cytoplasm, where protein synthesis is happening. Any questions on this before I go to how RNA transcription works?
Okay, Chris, if the anticodon is AUC, what would the codon be translated? Does it have to specifically bind AUC? Could it be like another start codon, or it's just AUC?
Yeah, so each codon pairs with only one anticodon. So AUC, it will have to bind with the complementary codon, which would be UAG.
UAG, okay.
Yep. Or GAU. It doesn't.
So is this the start? Is this the start, or is this, because you said it's going from the nucleic acid to the amino acid? So is this a start codon?
No, so I'll go over the start codons in a bit. So AUC is not the start codon.
It's not, okay.
But this is the anticodon anyway. So the start codon will be the mRNA codon, and that would be AUG. I'll go over it in the next few slides.
Okay.
Okay, so RNA transcription. So this is how we use the DNA in order to make RNA. So similar to DNA transcription, helicase unwinds the double-stranded DNA, and then topoisomerase prevents supercoils. But different to different to DNA transcription, RNA transcription doesn't need a primer. So in DNA transcription, you need an RNA primer. But since you're making RNA, you don't need a primer. RNA polymerase can bind to the promoter regions in DNA. This is aided by transcription factors. So just like DNA replication, the template or antisense strand is used to make the single-stranded mRNA. So it's good to note that the template strand is always antisense, and the mRNA that you make is identical to the coding or sense strand. So if you see a template or antisense strand, the mRNA will be the opposite base pair to that. So if the template has like an A, the mRNA would be a U, while if it was a G, then the mRNA would be a C. And then, so after the RNA is transcribed from the DNA, it's called heterogeneous nuclear RNA. So that's the primary transcript that's formed, but before it can leave the nucleus in order to be used in protein synthesis, it has to undergo a bunch of post-transcriptional modifications before it's ready. So that's when they edit the mRNA. So they'll splice it, they'll cut out unneeded regions in order to create a specific mRNA that creates a specific protein. And so another type of RNA is ribosomal RNA, or otherwise known as rRNA. So rRNA is also made in the nucleus, but it's made in the nucleolus. And so that this is the RNA that's being used in ribosomes. So RNA, it's used to make ribozymes, which is an enzyme that uses RNA instead of peptides. So it's an enzyme that instead of amino acids, it's made of RNA. And this RNA, it's used in ribosomes, and it's used to catalyze peptide bond formations. So it's actually connecting separate amino acids through peptide bonds to form the full protein. So ribosomes, they're made up of rRNAs and proteins, and they have 3 binding sites for the tRNA that holds the amino acid to make the protein. So the first site, it's called aminoacyl. Then other site is P or peptidyl, and then the last site is E exit. So ribosome has 3 different sites for the tRNA. Any questions on this before I go into a question? Okay. All right, I'll go into the next question. All right. I'm gonna start the poll. So which of the following statements is false regarding transcription? So RNA transcription in eukaryotic cells. Give everyone a few minutes for this. So I see there's a question in the chat. So the tRNA amino acyl complex, it's not it doesn't actually have a charge unless the amino acid has a charge, but it's called charge just because of the amino acid that's bounded to it. So it's charged as in like it's ready for protein synthesis. It can be used in the ribosome to add on to the amino acid chain. Yeah, sorry for the confusion if I didn't mention that. Yeah, if anyone else has any questions, just post in the chat, or you can unmute after I go over this question. I'll give everyone about 20 more seconds before I end the poll. Okay, I'm gonna end the poll now. Okay. So most people play choice A, and that's the correct answer. So transcription happens in the nucleus because the DNA is in the nucleus. So the transcription would have to take place in the nucleus because the RNA is made from the template DNA. So RNA polymerase does only use one side of the DNA to transcribe it. Transcribe it. And then so it does need RNA polymerase does need a sequence, specific sequence of DNA to bind, and then RNA does use uracil instead of thymine. So choice A is the best answer. Anyone confused by this question? Or any questions in general before I move on? Okay. All right, I'm gonna move on. If you have a question, you can put in the chat and Janelle can try to help you. So then I'm gonna go over RNA translation. So how is the RNA used to form proteins? So RNA translation, so first thing, initiation, the ribosome binds to the mRNA sequence that was transcribed. And like I said before, it brings it together with a charged amino acyl tRNA. So that's the tRNA that is bounded to a amino acid. And when it brings it together, so when it brings the mRNA and tRNA together, if the codon and the anticodon match up, it will add the amino acid to the growing chain of the polypeptide. So the first codon always has to be AUG. So AUG is a start codon. So the chain where the mRNA sequence where there's an AUG sequence is the start codon. So that's where the ribosome will start translating the mRNA into a polypeptide sequence. So RNA translation. So the next step is elongation. It's a 3-step cycle that's repeated for each amino acid added. So the ribosome, it moves in the 5' to 3' direction along the mRNA. And the protein is made from amino, or the N terminus to the carboxyl or C terminus. So the peptide bond that's formed, it's formed from amino to a carboxyl terminus. And that's why it's called peptide bond because of the amino carboxyl bond. So the peptide bond is formed by peptidyltransferase. So peptidyltransferase is the enzyme that forms the peptide bond between the two separate amino acids to form the polypeptide chain. So like I said before, there are 3 sites in a ribosome for the tRNA. So the A site, it holds the incoming amino acyl tRNA complex. So it holds the next amino acid that we added, and that's determined by the mRNA codon in the A site. So if the tRNA has a matching anticodon to the mRNA codon in the A site, then the peptide or the amino acid will be added to the polypeptide chain. And then the P site holds the tRNA connected to the polypeptide chain. So each time a amino acid is added onto the polypeptide, that tRNA will move into the P site, and that tRNA holds the growing peptide chain. And once the amino acid from the tRNA is added onto the polypeptide, the tRNA is moved from the the polypeptide is passed from the tRNA in the P site to the tRNA in the A site. So like I said before, each time a amino acid is added to the polypeptide, the tRNA will transfer the polypeptide to the next tRNA in the A site, and then that tRNA will be moved into the P site. And then the last site, the E site, that holds the inactivated or uncharged tRNA. So after the tRNA gives up the amino acid bonded to it, to the growing peptide chain, it'll be moved into the E site before exiting the ribosome. And then that tRNA that left, it can be reused again. So it just has to find the matching amino acid, the amino acid that matches the anticodon in the tRNA, but it has to be a specific amino acid, since each anticodon is only specified by one amino acid. And then last step, termination. So once a stop codon moves into the A site, so once the stop codon moves into A site, the release factor RF protein binds to the stop codon. So the stop codon, they don't specify any amino acid. So these stop codons don't have a amino acid correlated with them. So the mRNA, the stop codon, it will bind to a matching anticodon on the tRNA, but then the tRNA does not hold any amino acid. What happens is the release factor adds a water molecule to the polypeptide, and that's used to hydrolyze the tRNA from the polypeptide. So that releases the polypeptide from the tRNA in the P site, and then the polypeptide is complete after its release. And since the protein is formed, the ribosome subunits dissociate until they encounter another mRNA that's ready for protein synthesis. But before the polypeptide chain can properly function, it has to undergo post-translational processing. So things like chaperone proteins has to bind to it in order to fold it into the proper shape. So proteins, they need a specific shape in order to function. And chaperone proteins help that by allowing it to fold into its proper shape. And then some proteins also go cleavage events. So the polypeptide chain has to be cut down before it can be active. So a large inactive protein will be cleaved into a smaller protein, but that activates that protein. So the polypeptide chain will have to be cleaved by an enzyme in order to be activated. So that's RNA translation. Any questions on that before I go into proteins? You can put in the chat or just unmute. Okay, all right, I'll move on. So proteins, that's the molecule formed by RNA translation. Like I said, proteins are polypeptides formed by amino acids through peptide bonds by the ribosome. The template is initially DNA and then finally mRNA before it's turned into a protein by the ribosome. And proteins have a wide variety of functions in our body. So some of the functions are, it can be an enzyme that catalyze biochemical reactions. So they form enzymes that speed up reactions without being used up. They decrease the amount of energy needed for chemical reactions in order to speed up the reaction. Or they're peptide hormones that are used for chemical signaling over long distances. And examples of those are insulin and glucagon. So these are peptide hormones. I know most hormones are steroids, but there are a few peptide hormones that are used. And then proteins, a main function of them are structural proteins. So they give shape to the cells and they're used for movement. So proteins like actin and myosin, they're used for movement. They're in our muscle, they're used by the cell for movement. And so, like I said before, the proteins, it needs a specific shape in order to function. So when denaturation happens, it disrupts the protein shape and the protein loses its function because of that. So proteins, they need a specific shape in order to function in our body. And it can happen because of changes in temperature, pH, or specific chemicals called denaturants. So if a protein gets too hot or too cold, it'll lose its shape and lose function. If the pH is with it outside of its optimal range, it'll also undergo denaturation. But denaturation, it causes the proteins to lose its 3D shape. So it won't change the amino acid sequence. So the primary structure, which I'll go over in a bit, it doesn't lose the amino acid sequence created by the ribosomes. It just loses the shape it adopted after RNA translation. So all the post-translational modifications that it had in order to get to the proper shape, it loses that. But if the change that caused the denaturation is removed, it can regain its 3D shape again over time. So any questions on that before I go into another question? You can put it in the chat or unmute. Okay, all right, I'll put up the next question. So like I said before, the term denaturation, it refers to a change in structural characteristics, primarily due to, let me start the poll. Okay, I'll give everyone a few minutes for this. I'll give everyone like 20 more seconds if you want to put in an answer. Okay, so most people chose choice A, but the answer is actually choice C. So choice A, covalent bonds, those are like the peptide bonds in the primary structure of the protein. So like I said, the denaturation, it changes the 3D shape of the protein, which I'll go over in a bit. So 3D shape is actually formed by like hydrogen by hydrophobic and hydrophilic interactions. So those are actually non-covalent bonds. Choice A, the covalent bonds, it's actually the same answer as choice B. So covalent bonds are the primary structure of the proteins, while choice C, non-covalent bonds, they're the secondary and tertiary structure of the molecule. So those are the like hydrogen bonds, hydrophilic, hydrophobic interactions. And then choice D, so the chemicals, the toxic compounds, they don't actually bind to the protein. They just disrupt the 3D structure of it. So just the presence of the chemicals will disrupt the structure. They don't have to physically bind to the protein. I guess there was some confusion on this question. Yeah, it was like a little warm up for the part I'm going to go over now. I think someone put a question in the chat. Covalent bond. Yeah, so the covalent bond is the primary structure. So covalent bonds, they form the primary structure. So the actual amino acid sequence, it's connected by covalent bonds. So each amino acid is connected to the next, to the amino acids around it by covalent bonds. So the peptide bond that form the actual protein, the protein amino acid sequence, those are covalent bonds. And those are hard to break, a lot harder than the non-covalent bonds. So it requires a lot more work to break those covalent bonds, while denaturation, since after you denature it, it disrupts the non-covalent bonds, but since those bonds are relatively weak compared to the covalent bonds, the protein can regain its 3D shape if you remove whatever caused the denaturation. So if you move the temperature to its optimal temperature, or the pH to whatever pH it functions best at, it can regain its 3D shape. Any other questions on this? All right, I'm gonna move on to the next. So now I'm gonna actually go over the protein structure. So the primary structure, like you said, it's the actual identity of the protein. It's determined by the chain of amino acids linked together by the ribosome and predetermined by the mRNA that was transcribed. So like I said before, they're stabilized by the covalent peptide bonds between the adjacent amino acids. So the primary structure gives the actual identity of the protein. So different primary structures lead to different proteins. And then the secondary structure. So the secondary structure is formed by adopting the most energetically favorable arrangement of the primary structure. So the primary structure is linked by peptide bonds, but the secondary structure is mostly determined by hydrogen bonds. So hydrogen bonds between the different amino acids, they provide stability. They form these two structures on the right. So you see primary is just the link, the chain of amino acids, but then secondary, they form these alpha helices on the left, and then on the right are these beta pleated sheets. So these are formed because of the stability provided by the hydrogen bonds. So the carbonyl oxygens and the amide hydrogens of the different amino acid residues, they hydrogen bond in order to form these specific secondary structures. So the primary structure is just a chain, but that's not very energetically favorable. So they adopt these two secondary structures in order to form hydrogen bonds between them, and that provides more stability. And then the secondary structure can be disrupted by denaturation, but it can reform these secondary structures if the denaturant is removed. And then tertiary structure. So this tertiary structure also adds to the 3D shape. It is formed by hydrophilic and hydrophobic interactions between the R groups. So each amino acid has a different R group. That's like the side chain that's attached to it. The side chains, it may be hydrophilic, which in that case, it will be on the outside, and it will form hydrophilic interactions, while hydrophobic groups, they don't like water, so they'll be more likely be on the inside of the tertiary structure. So it's more likely to find hydrophobic R groups in the center of the protein, while the hydrophilic groups will be on the outside. And these are all determined by the R groups connected to the amino acids. So hydropolar and charged amino acids, they're more likely to be on the outside, while hydrophobic R groups will most likely be on the inside of the protein. And then another thing that adds to the 3D shape are disulfide bonds or disulfide bridges. So this is when two cysteines are oxidized to form cysteine. So two cysteines combine together in order to form a disulfide bridge, and that also adds to the 3D shape of the protein. And disulfide bridges, they can also be broken apart by denaturation, but then also reformed as well after denaturation, denaturant is removed. And then finally, the quaternary structure. So not many proteins have quaternary structure. This is only when the protein has more than one polypeptide chain. So this is when the protein has multiple polypeptide chains combined together into a quaternary structure. So this is formed by aggregating the different protein subunits into a quaternary structure. So like hemoglobin would be an example where there's multiple subunits that combine together in order to form the full protein. Any questions on this, on the different protein structures? Yep, Syed, yeah, like I said, hemoglobin is like the classic example of the quaternary structure. This is, they actually do this to like save time and energy. So instead of making like a really large protein that has just one subunit, it'll create 4 identical subunits. So you can create four identical subunits that just require one specific sequence. You just make it 4 times and then combine it together. So you can do that like 4 times as fast technically, since you can create 4 single subunits at once and then just combine it together into a full coordinary structure. All right, if no one has any questions, I'll move on to the question. So let's see if you guys are paying attention. Which level of protein structure is stabilized primarily by hydrogen bonding? Let me start the poll. Okay, I'll give everyone a few minutes on this. All right, I'll give everyone a little bit more time. I see a question in the chat. What pushes protein formation in terms of energy? Do you mean like protein formation? It's usually driven by like transcription factors. It's like pretty complicated, but transcription factors, they'll push to make a certain protein. I guess what makes yeah, so usually the energy from ATP, I believe, so the nucleotides, they'll have the phosphates binded to it. That usually is used to form the mRNA, and then the need for specific proteins, they'll use energy to make the protein. All right, let me stop the poll. Okay, most people chose choice D, and that's the correct answer. So yeah, the secondary structures, the alpha helices and the beta pleated sheets, that's driven by hydrogen bonding. So H bonds between the carbonyls and the amides, those drive the protein to form those specific secondary structures. Any confusion or questions on this problem? All right, yeah, so that's all I have for everyone today. Thank you guys for coming. If you have any questions or anything you want me to go over again, just put it in the chat or unmute. All right, I'm gonna stop the recording. So,